Thank you very much for the organizers for inviting me and that introduction. My name is Eddie Sweeterman. I'm an assistant professor at Earth and Planetary Sciences Department at UC Riverside. I'm going to talk about prospects for biosignatures, which is going to be a mix of a review of biosignatures, frameworks for understanding those biosignatures in context, and then prospects for actually detecting them and how they would manifest observationally. Okay, thank you very much. So a big question is, what is the prevalence of life in the universe? And astrobiology is a study of the origin, evolution, distribution, and future of life in the universe. Of course, most of the universe is outside of our solar system. And so if we want to understand the prevalence of life in the universe, we're going to have to look at exoplanets. Of course, we cannot go there. So we're going to have to think about remote observations of those exoplanets. This is a civilizational goal. It's not going to be an easy thing to do. It's bigger than one observation or one set of observations or even one mission. We're aided in this quest by the observation that life has left an indelible impact on Earth and its atmosphere. The oxygen we breathe is from bacteria and algae and plants. We have methane in the atmosphere produced by methanogens. Even the nitrogen and carbon in our atmosphere is cycled many hundreds to thousands of times faster by life than it is by geological processes. Life and the atmosphere are inextricably linked. It's not the case that Earth is some abiotic background in which the influence of life is superimposed. The atmosphere can't really be understood if you don't understand the influence of life. The composition of our atmosphere through both planetary and biotic factors isn't something that can be described in, say, like a metallicity. There's no layer of the atmosphere that's at thermodynamic equilibrium that's repartitioning the molecules. For example, we have a nitrogen-dominated atmosphere, even though through abundances on Earth, nitrogen is the 30th most abundant element. Yttrium is more common on Earth than nitrogen. This atmosphere has changed drastically through time, through the coevolution with life. And those changes are not necessarily predictable on first principles. They depend on evolutionarily contingent events like the evolution of oxygenic photosynthesis. And it's really hard to describe Earth's atmosphere with an abiotic model. If you did, that abiotic model would be wrong. So if an alien were to build a Habitable Worlds Observatory and observe Earth and conclude, because they have very strong priors against life, that it's actually some abiotic process producing the combination of gases in our atmosphere, they would be wrong. And those aliens should feel bad because they have given up the chance of finding life. And also, philosophically, is it really any better to misattribute an abiotic process to a biological planet than a biological process to an abiotic planet? Both would be bad, right? And so this is to say, even if it's going to be hard to confirm life, and it might be, life's going to mess up your planet's spectrum. And so the possibility of life cannot be excluded a priori. This chart here shows a, see if I can get the, oops, pointer. I don't think I can. Oh, there we go. Yeah, the fluxes of gases with life and without life. And you can see those are very different. And of course, we're looking for a planet in which this is true. We're not going to be able to find extremophiles clinging to life in the subsurface of an exoplanet. So the question really isn't how prevalent is life in the universe. It's how common are planetary biospheres like Earth in the solar neighborhood. That's actually a question we have a chance of answering. So what is a biosignature? In the context of exoplanets, we say it's a remote indicator of life. In all cases, when we say biosignature, we really mean potential biosignature because we're not going to put an asterisk after it all the time. There are molecules and features and substances that we are looking for that are produced by life. All of them have potentially abiotic sources, and it's always going to depend on context. When we're looking for, say, a molecule or a substance generated by life, we want it to, fit some criteria. It probably needs to be global in extent because we're getting the whole disk of the planet. If we're, say, direct imaging the planet, or getting the entire annulus of the atmosphere if we're looking in transit, we need to look for things that actually produce line absorption or other spectral features so that we can detect them remotely. They need to be robust to photochemistry, build up to detectable amounts. And they need to be, given context, separable from abiotic processes. And there are a couple of ways of going about this. So one is by analogy. So we can think about Earth and the things that are in Earth's atmosphere or surface that are made by life. It automatically passes these tests, because if we look at Earth as an exoplanet from say within the solar system or from low Earth orbit, we can confirm what's observable and what's not. There's a bunch of models and data that have been developed for Earth that we can use. They already exist. We're always going to have more information about Earth, and we have Earth through time. The limitations here is that Earth is not probably the only example of a habitable and inhabited planet. And so we're missing out on all the different tracks a living planet can take. And so we might think about agnostic and other approaches, thinking about looking for, say, thermodynamic or kinetic disequilibrium in atmosphere, very different on a terrestrial planet because there is no layer that's hot and in equilibrium and dredging molecules up. It's a lot more complicated with surface boundary conditions, et cetera. And so we can think about these more agnostic frameworks for detecting life. The disadvantage, of course, is that we still have the false positive problem, and also we need to do a lot more modeling and laboratory work. But we get a broader universe of signatures. I think that there's a lot of in-between area where we have things that are made by life on Earth today that maybe don't build up to detectable amounts, but we could imagine that being the case on another planet. So what are some possible biosignatures on terrestrial planets? So I kind of split these into three categories because they depend on basically the observational manifestation of these signatures. So they're atmospheric gases. We talked about oxygen as a product of photosynthesis on Earth, at least, made by, say, cyanobacteria. One photochemical byproduct of that oxygen is ozone. They have different absorption features and different wavelength ranges. So depending on what kind of observation we're making, we could look for one or both of those gases. Methane on Earth is made by methanogens. Of course, methane is found in most of the atmospheres of the solar system from an abiotic sense. I'll talk about why it's maybe a biosignature on Earth and not in the other places. And then many other gases that have trace amounts in Earth's atmosphere are made by life and maybe not as easily confused. There are surface signatures like the vegetation red edge and other pigments and temporal changes to these things. That's correct, requires a time element and may be very hard. And so there have been many reviews on biosignatures. I've listed just a subset of these here, and you can check those out. We had in 2018 a review at the time of everything we knew about biosignatures, and this is one of the graphics from that special issue in Astrobiology. What does life produce? Can a dead planet fool us? How do we interpret limited data? All really important and illustrates the interdisciplinarity needed to address the question of life elsewhere. So this is a model of Earth spectrum. This is, basically an albedo spectrum of Earth, geometric albedos comparing to, a perfectly diffusing disk. You don't really need to worry about that. But what it's showing you are the features that are large in Earth's reflected light spectrum. That is light from the star that's scattered or reflected by the atmosphere, clouds, and surface of the Earth. And so some features you can see, Rayleigh scattering tail, that kind of gives us an idea of how massive our atmosphere is. You can see here, the Hartley ozone feature. It's a huge feature. That's our ozone layer. So we're seeing the observational manifestation of our ozone layer. This is the big one that people talk about, the oxygen A band, wanting to look for that. And of course, we have many water features, teeny methane for Earth, modern Earth at least, a little bit of CO2. Of course, that's not actually how we would get that data. We have had many talks before talking about how challenging it is to get an Earth around a sun-like star. It's outshone by the star. The planet is outshone by the star by a factor of 10 billion. So we need this 10 to the negative 10 contrast. So here's a, apologies, a theoretical, a model of a direct imaging observation of the solar system from 10 parsecs. And you can see the Earth is this blue dot here. And if we were to run it through a spectrometer, we would get this kind of data here. Of course, we'd have actual error bars. The spectral resolution at different wavelength ranges would be different because of the needs of the instrumentation at those wavelengths. So we'd have coarser resolution in the UV and near infrared than in the optical. But you can see even with that, those limitations, we can get things like the oxygen, the ozone, and the water on a modern Earth-like planet. So this is really what we're going to go for. And Sean is going to give a talk later this afternoon about observatory UV, vis, optical, like infrared, like HWO. And so retrievals will then help us confirm signatures. So we're going to look for the features and hopefully put those through retrieval models, and we can obtain the abundances of things like oxygen, water, and ozone. And we hope to do this with about 25 targets with HWO, according to the 2020 Astronomy Astrophysics Decadal. That's the goal. And the reason why we want to do 25 is because that actually gives us a statistical sense for the prevalence of these signatures on exoplanets. So if we found none, then actually that tells us something about the nature of the distribution of life in the universe. It's a meaningful result either way. Of course, I mentioned that Earth's atmosphere has changed through time. Earth represents not just one planet, but a palette of plausible biogeochemical states for a planet. So this is a plot that just shows the evolution reconstructed through geochemical processes, the evolution of the oxygen in our atmosphere. So 2.5 billion years ago, there was no oxygen in Earth's atmosphere. About 2.33 billion years ago, there was an event called the Great Oxidation Event, where the amount of oxygen in the atmosphere drastically increased. It still wasn't as high as modern day until the last several hundred million years. It's no coincidence, of course, that we inhabit this time period because we needed that oxygen in order to have our aerobic respiration be sufficiently powerful enough to support large macroscopic creatures like us. And of course, other things probably changed through time for the same reasons. The methane in our atmosphere, we suspect the carbon dioxide in our atmosphere has also changed as a result of the carbonate silicate cycle and the influence of life. And so we can think, the Earth has changed through time. We're not just looking for Earth 2.0. We might be looking for Archean Earth 2.0. We might be looking for Proterozoic Earth 2.0. And so these are plots that show different spectra of these hypothetical reconstructions of Earth through time. The reason why these lines are different depending on the star is because these models by Sarah Rugheimer assumed self-consistent photochemistry. And of course, the photochemical, the spectral influence of the star will control the lifetime of the gases, which then will control the manifestation of their spectral signatures. This is a synthetic mid-infrared emitted light spectrum of the planet through time. And then this is a optical near infrared UV synthetic transmission spectrum of the Earth through time. And there are many different authors who have done this and thought about what it would the different signatures and biosignatures be of Earth through time. And I've just listed a few of those here. And this has made it into the reports. So the LUVOIR final report, the HABEX report, and even the Decadal, I believe, all understood and included the fact that Earth has evolved through time. So if we're really thinking about studying a habitable and inhabited world, we've got to utilize the richness of information that the evolution of our own planet provides us. Earth is always going to be the best studied planet, and we're always going to be able to get more information about the evolution of our own planet and the other planets in the solar system, of course, than any exoplanet. Of course, Earth doesn't necessarily trace out all of the possible manifestations of biosignatures on planets. And we have to worry about things like false positives. So false positives are abiotic processes that produce the substances or gases that we'd otherwise attribute to biology. And so here's a figure from one of Vicki Meadows' papers, and it shows all of these different ways that folks have determined that we may get abiotic oxygen on otherwise habitable planets or planets in the habitable zone or temperate zone. It used to be thought, I guess, in the era of TPF that, it would be very unlikely to get oxygen in the habitable zone. And we've just had a myriad of papers in the last 10, 15 years that have kind of disputed this and thought up all these scenarios. So one is the photolysis of a CO2 rich atmosphere. You can get some oxygen that way. We have a little bit of an example on this on Mars, where there is a little bit of oxygen and ozone because of the dryness of the atmosphere, the fact that those two species have a hard time recatalyzing back into CO2 due to a deficit of catalysts. So you can imagine supercharging this on an M-dwarf planet with a high FUV flux. You can also imagine scenarios where you go through a one-way greenhouse and you exhaust all your oxygen sinks and you're left with abiotic oxygen that's built up. In any of these cases, though, you'd also predict corresponding spectral signatures or the absence of other signatures. So you might expect if you're have a massive abiotic oxygen atmosphere, you actually have too much oxygen and you get these O2 CIA bands, collision-induced absorption bands, that you wouldn't get in a sort of a biological atmosphere that's only 0.2 bars of oxygen. If you're getting your oxygen through the photolysis of CO2, you should see a lot of CO2 and CO. And so observationally, you might be able to distinguish between the true positives and the false positives. And of course, on Earth, it's really the combination of things that tell us about life. Folks have done the same thing with methane. And really the idea for methane is on Earth, the photochemical lifetime of methane is very short. It's about 10 years. And so even though methane is present, of course, in all the gas giant planets and Titan, the fact that it's on Earth in any concentration is surprising. It requires a large production flux of that methane. And so here's a figure from a paper by Maggie Thompson and Josh Sanson-Totten about the context of methane as a biosignature on terrestrial planets. You've probably heard of this paper. We're at the Sagan workshop. There's a paper by Carl Sagan in 1993. They took the Galileo spacecraft, which did a flyby of Earth to get a gravitational boost on its way to the Jupiter system. And they looked at Earth and wrote this paper with the idea that we're going to assume we don't know much about this planet except for the information we get from the instruments on Galileo, including the spectrometer. And what they did is they looked at the different gases that are present in Earth's atmosphere, oxygen, nitrous oxide, methane, and then they calculated how far out of thermodynamic equilibrium are these gases. And so here are the results. I want you to look at one number right here because we're going to recalculate it, this 10 to the negative 145. So this is a really low number. It's much less than one molecule in the entire atmosphere. And it tells you maybe in first pass that something weird is going on. There's some caveats in thermodynamic equilibrium that I'll mention to you. But first, we're going to reproduce this calculation in just a second. So the calculation of, say, the Gibbs free energy that's available in the atmosphere has been done in the context of thinking about biosignature. So on Earth today, there's a small disequilibrium between oxygen and methane. The reason why it's small is because there's a small amount of methane. There's a huge disequilibrium between the nitrogen, the oxygen, and the water. The nitrogen should be in the form of nitrates dissolved in the ocean. And so this is a paper by Josh Sanson-Totten that did these calculations and showing that if you could characterize the surface and the atmosphere, you'd calculate a much larger disequilibrium than just through the atmosphere. It's been done for Archean Earth. So in Archean Earth, the disequilibrium instead exists between the different oxidation states of carbon. So methane and CO2 are in a strong disequilibrium. And then just recently, this preprint is available. It's just accepted paper by Amber Young, did the same kind of disequilibrium calculation for Proterozoic Earth and showed that you can indeed, for some cases, for some possible reconstructions of Proterozoic Earth, retrieve the actual Gibbs free energy disequilibrium, atmospheric disequilibrium of Proterozoic Earth. So this is all interesting, but there are some caveats. So what we did was we calculated the thermodynamic disequilibrium of the atmosphere. And in particular, for the Sagan calculation, it's between methane and oxygen. And the net reaction is methane and oxygen react to form carbon dioxide and water. Okay, so you can do this calculation, the Gibbs free energy, okay, of the products minus reactants. Okay, if it's negative, there's it's a spontaneous reaction. So you can do this with the numbers for methane and oxygen and CO2 and H2O. And you can calculate, how far out of disequilibrium the methane is. And so if you were to assume that Earth's atmosphere were in equilibrium, you would anticipate that the concentration of methane was this small. Okay, so what is one caveat here? What happens at room temperature if you put oxygen and methane in a box? Nothing. Nothing happens. So that's not actually how the methane on Earth is consumed. It's consumed through photochemical reactions. The biggest sink for methane in Earth's atmosphere is a hydroxyl radical, which was produced through ozone photochemistry, actually. And so you can calculate the lifetime of that methane, assuming that its primary sink is hydroxyl, and you get a lifetime of 12 years. So the difference is that the thermodynamic disequilibria doesn't tell you the time scale of the disequilibrium. So you could have a single event that creates it. What you really want to do is understand the lifetime and the flux, because that tells you maybe something about the surface biosphere. And so this is a plot also from Maggie Thompson's paper that compares the amount of flux you would retrieve, amount of methane flux on Earth from the surface and abiotic sources you'd expect from a terrestrial planet. And you can see they're separated, and this is a logarithmic plot. And so really what we want to do is retrieve on the flux of the gases. And that may tell us more about, is this consistent with production from, say, a global photosynthetic biosphere? Importantly, the lifetime of the gas is going to vary drastically based on the star. And so this is just a set of simulations showing nitrous oxide flux, nitrous oxide concentration versus flux in atmospheric O2. And you can see that between these two stars, there's a huge difference. And so therefore, you would get a different answer if you were to retrieve on the flux. This cuts both ways. We want higher concentrations because they're more detectable, but then if the lifetime is high enough, small abiotic sources can generate the same signature. So we kind of want a compromise where if we see the gas can build up, but if we see the gas, it indicates a very large surface flux. And so folks have done this. There are different gases that we can think of. Even gases that are made in equilibrium in giant atmospheres can be in strong disequilibrium in terrestrial atmospheres. And then there are gases that are made by life as metabolic products, like isoprene and nitrous oxide, that are not found, at high temperature, high pressure layers in gas giant atmospheres, but could, with a high enough biological production flux, build up to detectable layers, detectable levels. Of course, all of these things have small abiotic sources, and they have to be considered. The best near-term target is probably something like the TRAPPIST-1 system, for a couple reasons that have to do, of course, with the fact that we're going to have the best signal to noise for TRAPPIST-1, and also we're aided by this flux abundance relationship. So even Earth amounts of methane can generate large methane bands. So if you were to put just the same Earth around TRAPPIST-1, you would assume, given the same methane production flux, a much larger concentration of methane by a factor of 100 to 1,000. And that, of course, would correspond to bigger transmission features. There's a paper by Josh Chrysanthi-Daughton that does this for the Archean Earth and shows you could retrieve the methane CO2 disequilibrium. We saw one of these plots by Matteo Brogi in an earlier talk that showed that finding some of these things is going to be challenging from the ground, particularly oxygen. However, finding things like methane and CO2 may be much easier from the ground, due in part to an absence of as much telluric interference. And so you can imagine that there's a complementarity between ground-based observations, which could maybe start earlier, and future space-based observations. We want to look in multiple wavelength ranges. So I showed you reflected light spectrum of Earth, and we're going to hear from Sean. We also heard from Sasha about the mid-infrared and the LIFE mission. The advantage to the mid-infrared is that molecules have a lot of opacity in the mid-infrared. And so we have just access to different molecules and different abundance ranges of those molecules if we look in the infrared. And then of course, we don't want to forget that if we want to look at surface biosignatures, we need reflected light. So I'm just going to mention that surface biosignatures are not necessarily exactly like Earth. There's a vegetation red edge, but there's also all these other pigments. We could imagine them being more higher manifestation on other planets. We need more laboratory data. Even things like the near UV cutoff of water vapor absorption aren't as well known as they can be. And there are many gases made by life that have no opacities that we know of. So that's it. Thank you.
Okay, we have time for a couple of questions. One right here.
Hi, quick question. In terms of actually detecting these signatures, it seems like the signatures we'll be able to detect, will be in the atmosphere. Does that mean that we would be unable to detect signs of subsurface life? Are we really only constrained to surface life searches with exoplanets?
Yeah, I mean, so the real answer is you want to have a robust exchange of gases with the atmosphere, and you're also going to be limited by the size of your biosphere, which is going to be in some way related to the abundance of the signatures you're trying to detect. We think that we're probably going to need photosynthesis to power a biosphere that's large enough to be detectable. And then the other thing for like subsurface life is they're usually chemosynthesizers. They're destroying equilibrium rather than creating it. So it might be hard to see it. We might have false negatives for subsurface biospheres.
Hi, so you mentioned using like seasonality and like temporal changes to as a biosignature. With that in particular, how difficult do you think it'll be to separate that from just the planet being variable and things that are like complete abiotic processes?
Yeah, difficult. But it's it goes to the question of, what do we do after we get Earth 2.0? And once we find it, we're never going to forget about that planet, right? So it's going to go to okay, we found Earth 2.0. Now we're going to think about characterizing that planet. So if we find a biosphere, let's characterize that biosphere. And so you imagine, again, I mentioned this is a civilizational goal. I don't think we're ever going to just hang our coat up and say, great, we found a biosignature, we're done.
Okay, we have, yeah. So this online question is asking, how dependent is our understanding of biosignature searches on the assumption of tidal locking? Would a non-tidally locked planet show significantly different spectra?
So great, I have a backup slide for that. So yeah, I mean, a lot of assumptions, of course, a lot of the modeling is done using 1D, and of course, we're informed by the actual Earth, which is a rapid rotator. If we had a tidally locked planet, that affects the distribution of gases, possibly in the atmosphere, because you have the photolysis going on the day side and possibly a reservoir that protects you from photochemical reactions on the night side. Howard Chen had a paper here where they found that standard biosignatures like nitrous oxide and methane only really differed by about 20% compared to the 1D prediction, so that's good. But other gases with a much shorter lifetime, like dimethyl sulfide, could have a significant difference between the day and the night side. And so that may have a big observational effect.
Stop it there. Let's